All right, so let's start off with glycolysis. And I'll just put all of the major processes in a little box to separate it. And let's just give some fun facts about glycolysis. Where is it going to happen? And is it aerobic or anaerobic? So first we know this is going to happen in the cytoplasm. And it's going to be anaerobic. We know when we start glycolysis, we're starting with the 6-carbon molecule called glucose. So I'm just going to draw six little circles, label glucose. And we know glycolysis is a many-step process, but I'm just going to kind of simplify it into an investment and a cash-out phase. So your first phase is investment. Remember, we invest 2 ATP, and I'll depict that here. So I'll draw ATP. When you expend ATP, you get ADP. And so then, if we're plug in 2 phosphates, we'll draw these attached to the phosphates. Then a few more things happen, which we don't need to know the details of. And we get these 3-carbon molecules called pyruvate. Once I finish this, I'll slow down so you guys can catch up. So an important thing to note here is that we're taking 2 NAD plus 4 ADP, and from that we are generating 2 NADH plus 4 ATP. And I'll label this as pyruvate. Okay, I'll slow down a little bit so you guys can catch up. Maybe what I'll do is the key terms that we need to know, I'll highlight. Cytoplasm, we need to know that glycolysis happens in the cytoplasm, and we need to know that it's anaerobic, not aerobic, anaerobic. And then an important product, ooh, smeared it a little bit. We got pyruvate. All right, so that's glycolysis. So we'll separate this with like a dotted line or something. Okay. So pyruvate, we're assuming aerobic conditions. We're going to feed this into another process called pyruvate oxidation. Remember, this is going to happen in the matrix of your mitochondria, and this is the first aerobic phase. Now, we don't necessarily consider it aerobic because it's using up oxygen, because in this phase we're not necessarily using up oxygen, but it will not happen unless it is in the presence of oxygen, or it will not happen unless there is oxygen present. Okay, so we're taking these 3-carbon molecules here, and it's a pretty short process. What we're doing is we're taking 2 coenzyme A, we're adding it, and then we're taking 2 NAD plus to make 2NADH. And we know that another product of this is CO2. Okay. So we get sorry. So these are acetyl-CoA. Sorry, it's a little cluttered over here. And I'll mark this as acetyl-CoA. And I'll even put a little asterisk to, so CoA equals coenzyme A. So it happens in the matrix, and it's aerobic. And the product that's moving on is your acetyl-CoA. So pyruvate is moving on, acetyl-CoA is going to be moving on. Oops, sorry. All right. You guys good so far? Cool. All right. So what's next? We've got the citric acid cycle. So what I'll do is I'll just draw an arrow here. So your acetyl-CoA. I'll just draw CoA here. And so remember, we start off with a 1, 2, 3, 4 carbon molecule called oxaloacetate. Which moves into become a 1, 2, 3, 4, 5, 6 carbon molecule called citrate. Oxaloacetate and citrate. And what happens in the citric acid cycle? Well, it's a cycle. So many reactions happen, you shuffle stuff around. So I'll spare you the details of all the other stuff. Just know acetyl-CoA is being plugged into oxaloacetate to make citrate. And let's label this citric acid cycle. Citric acid cycle is going to happen in the matrix of the mitochondria, and it's going to be aerobic. Now let's talk about what we're making. We got a lot of room here. Okay. So I'm gonna be detailed here, but I'll tell you what the most important things for you to know here. So we take 6NAD plus, we take 2FAD, we take 2ADP. We plug that in, not all in one step. This is in various steps, but I'm just summarizing it here. And ooh, this is hard with the mic right here. The mic's like right here. Okay. So as a result, we get 6NADH, 2FADH2, 2ATP. And remember, our citric acid cycle is the heaviest producer of CO2. I think we get 4. I think we get 4 CO2. Yes, we get 4 CO2 out of here. Okay, so we don't need to know that you're getting 6 NADH. Just know that you're getting the most NADH. We don't need to know that we're getting 2 FADH. We just need to know that the only thing that out of all of these steps so far that are going to produce FADH is the citric acid cycle. 2 ATP, that should be easy to remember. You're getting 2 ATP, you're getting 2 net ATP from glycolysis. Remember, you're not getting any ATP from pyruvate oxidation. And then also know that citric acid cycle is the highest producer of CO2. But we don't need to know the details or the numbers, the specific numbers. Okay, so that's the citric acid cycle. Let me highlight the important stuff, or at least that stuff. All right, now after that we've got our electron transport chain. So we're going to be moving this direction. Let me draw the. Actually, let me start it first before. Okay, so we've got our electron transport chain. I'm going to really simplify the phospholipid bilayer. So we're going to pretend like this is one phospholipid bilayer. And then we got, remember we have our inner membrane and our outer membrane. So this is, or we'll say that this is the cytoplasm. And this is the matrix of the mitochondria. And this is going to be your intermembrane space. And I already messed up. One sec. I need some room for the enzymes that are involved. Oh, okay. Okay. Now we got this right here, and then we got big guy over here. Okay, I'm just making space for the proteins that are the intermembrane proteins. So sorry if I messed up your guys' drawings. So we got one right here. We've got one inside of here. We got one that kind of like hangs out here. And then we got another. And I'm being thorough for the sake of being thorough, but you guys don't need to know like the shapes and the locations or even the names of these guys. I'm just because I don't want to give false information if you guys were to look up these notes like some years from now and say, oh hey, he did a bad drawing. Okay. All right, so now I'm going to go back. I'm going to go back to the top, and I'm going to circle all of your electron carriers. Oh, sorry. Okay. So I circled all of the electron carriers from the top, and they're all being brought to the matrix of the mitochondria, in which they drop off their electrons. So I'll just draw a little electron here. This drops off an electron here. And then, remember, NADH drops off its protons. Same with FADH2. And so, remember like I said earlier, this electron really wants to move to this protein here. So it's going to bounce from protein to protein to protein. And we know that its last stop, it wants to move over to, it wants to combine with oxygen. We'll just say O and 2 hydrogens, and we'll just say that these all combine together to make H2O. It's one of your products. So remember, this is the electron moving from protein to protein. Now we know that in the process of this, we're using this kinetic energy to pump hydrogens into the intermembrane space. Same with here, we got another hydrogen moving into the intermembrane space. And so over time, you're generating a very acidic environment in your intermembrane space. This guy right here, I'll color him blue because he's special. So, and I'll label this ATP synthase. Okay. So all of your electron carriers bring their electrons to the electron transport chain. I'll write it here, electron transport chain. Sometimes we just use ETC to denote it. So electron transport chain, here's your intermembrane space with all of your hydrogens in there. You generate that by converting this kinetic energy of the electron movement through the from protein to protein. And then what ATP synthase does is it allows the movement of your hydrogen to go through, but at the cost of making some ATP from ADP. So ADP, ATP. And we could say around 36 ATP. It's important to know that H2O is made, but it's also important to know that oxygen is used. Oh, I'll keep it up here. I guess citrate's an important term to know. Same with oxaloacetate. All right, so we finished all of the aerobic chain. Oh, you know, something I didn't mention about the electron transport chain is that this is happening in the matrix. We could even just piggyback off this and say it's aerobic, right? Just to be thorough. Remember, ATP synthase is the enzyme that's going to catalyze the conversion of ADP into ATP. Put some on the screen here. Okay, cool. All right, last step. So we're done with the electron transport chain. That's the whole aerobic process. So we're going to move up here because it's more closely tied to glycolysis. So what's next is fermentation. And we know that this is going to happen in our cytoplasm. And we know this is going to be anaerobic. And it's a quickie. So let's just highlight, one of our pyruvate molecules, because we know that's what we're using for it. And so what we're doing is we're just, in humans, we'll just say in humans we're making lactate. Some people call it lactic acid. And we're taking NADH, to be more specific, to NADH, and we're making 2 NAD+. And what we're doing here with this NAD+ is we are plugging that back in to glycolysis at this step here. So what I'll just put as a note here, creates NAD+. Now, could anyone say what's happening to this NADH? Is it becoming oxidized or reduced? This one right here, this NADH in fermentation. Oxidized or reduced? Oxidized, yeah. You're going from a big thing to a slightly smaller thing. Or you can go from a thing with more electrons to a thing with less electrons. Cool.